Yes. Okay. I think we can. Yeah, we can welcome Jude.
Can you guys hear me?
Yes. Yes.
Yes. Yes. And you can see my screen? Cool.
Yes.
Well, first, I just want to say thank you to Boriana and Heidi for inviting me to speak and talk about my work. And thank you to everyone who's joined this Zoom mixer. This is an interesting format, but I'm into it. So my name is Jude Abuzaina. As Boriana mentioned, I'm Palestinian Canadian, and I'm currently pursuing my PhD at Rensselaer Polytechnic Institute in upstate New York. But I'm sure everyone's lives have shifted because of COVID. So currently I'm speaking to everyone from back home in Canada, which is kind of nice. So I figured I'd do a quick run through just some of my practice, how I approach my work as an artist who's interested and curious about the sciences, and just talk about some past projects and upcoming works. And, if anyone's got some interesting articles or things they want to send along the way, that would be really cool. So as someone who has spent their first half of their life in the Middle East and then later immigrated to North America and Canada, I sort of exist in this in-between space, this duality of identity as a Palestinian, but also as a Canadian. And it's in this in-betweenness, this duality of identity and the tensions of that in-between space is where I like to situate a lot of my work. So my practice usually begins in the realm of social engagement. I usually start by hosting a traditional Palestinian meal for guests, friends, family, colleagues, people in the communities that I'm inhabiting. And I make a spread of traditional Palestinian food. And I think of the dinner table as a site for alternate discussion, the site of gathering to discuss and bring to light different narratives that are not shared in mainstream media around Palestinian identity, culture, the political conditions that currently exist and that have been ongoing for the last 70 plus years when we're talking about tensions in the Middle East, but also specifically with Palestinian-Israeli conflict. And so food becomes this really beautiful vessel and vehicle to talk around identity politics and to bring forward a lot of these alternate discussions. And so that's sort of like step one of how I really bring people into the conversation, and food becomes this really great vehicle to talk about a lot of these things. So you can see here, this is some documentation from one of the first gatherings that I started hosting. And more recently, Matluba has become the center, the source material for a lot of my work, a lot of the way that I approach creating my artwork. And Matluba, I'm obviously biased because this is also my favorite Palestinian dish to make and to eat. And so Matluba is this, the best way I can describe it in North American culinary terms is like a one-pot casserole. And you combine all of these ingredients that at face value you don't think that they go well together, but once they're all layered in this pot, you can see on the picture on the left all the different layers of the ingredients that come together with the spiced rice that combine and create this very sculptural looking rice dish once it's all cooked and the pot is removed. And so this has become like my main source material for my artwork. So once I'm done hosting these meals and Matluba's the star of the conversation and the food that we're sharing, I usually take the leftovers and you'll see what happens in terms of turning that into artwork. Here is some documentation from some other meals that I've hosted during a residency in São Luís in Portugal. And this was a really beautiful moment where there also started to become this conversation, this cross-cultural conversation between food, where we have a spread of very traditional foods. In this case, there's some Portuguese food and Palestinian food, and something really harmonious starts to happen there in terms of the dialogue. the meal that's presented, and just this like really beautiful cultural experience and cultural exchange. And so then I'm able to take the leftovers from all of these meals, and they make their way into Petri dishes and into the lab space where I tend to produce a lot of my work. So here you can see that there's leftovers from matluba. There's some rice grains, some cauliflower, some tomatoes. Sometimes I just sort of throw the ingredients in there and let them do whatever they want and document the growth and decay over time. In other cases, I'm a little more meticulous and I'm spelling out words, or I'm creating images or drawing with them, so to speak. And over time, I document the growth and decay, the different cultural aspects that begin to form, making the invisible visible. And metaphorically, I'm thinking a lot about the growth and decay of also like capital C culture in this case. And from these still images of the Petri dishes, from all of these like beautiful blooms and, you know, things that exist within the natural scape of the Petri dish, I'm able to animate these images into moving images and create these kaleidoscopic videos that really engulf the body, and playing with projections, playing with site-specific installations. And I'll show a little video clip of what these videos tend to look like. So you can see here, again, everything that you see in these images, the source material is what you saw previously in the Petri dishes with matluba. So this is all traditional Palestinian food that's been manipulated with mirroring, fractals, but that's really all I'm doing. I'm not changing the colors. I'm not manipulating anything else besides reflecting and refracting the image on itself many times. And I'm sort of doing this as an ode to the kaleidoscope, Islamic art, sacred geometry, a lot of these aesthetic influences that I really grew up around and that are very prominent in the Middle East. I'm also interested in this idea of preservation. Again, when thinking about culture in terms of erasure, growth, and all of these different tensions that exist in this like dual existence, and sort of like the in-betweenness of existing from more than one culture. So here I'm also taking some leftovers and I'm encasing them in resin and sort of playing around with what this idea of preservation might look like. What does it mean to preserve something in that particular moment in time, especially when it has a livelihood? I'm also very interested in playing with different technologies. So I combine both like new media and old media, new and old tech, in a number of different ways in both how I work in the lab space versus my kitchen and at home, when I'm working with different technologies and creating my artwork, moving between digital and analog. And I like to do that too, because I'm also echoing a lot of the different strategies that women use in migratory kitchens when you're moving from one place to another, when you're adapting and conforming to a number of different ways of existing. You're usually moving back and forth between a lot of different things that you know and don't know. And that's sort of how I approach my way through the lab, because first and foremost, I'm an artist, not a scientist. And so I often find myself approaching lab technologies and like being in the lab space in the same vein as when I'm in the kitchen, how I follow a recipe for, you know, making a meal, or, you know, moving through adding different ingredients to get from point A to point B, and so on. And these are just some still images from the videos, the kaleidoscopic videos that I'm able to make. And so I'm able to extract these still images of the matluba in its like larger kaleidoscopic form. And I can show my work in a number of different ways. So there's the still images that I can print in a number of different formats. I create different video installations with new and old technology, with projections. So here you can see like more traditional prints. This is Kathy High's work next to mine. I love this photo so much. it also makes me laugh because here's my work in the background. It's like this very traditional Palestinian food that's been like presented in this contemporary way. And in the forefront, there's like poop. And this is an installation that again, I'm biased. This is my own work, but I really love this work. This piece, I've taken some of the patterns that you saw earlier and they were printed on these really huge sheer textile, and I was able to do a site-specific installation at the Ontario Science Centre in Toronto, where I was thinking about like domesticating this very brutalist cold space. And again, thinking about ideals of home. What does it mean to exist in this in-between space as an immigrant, as a refugee, to move from one place to another? And what are the strategies that we take to make a space feel more like home? And for me, it really boils down to like the domesticity of a place in line with food. And so this was sort of my way of intervening in that space. Then I'm also able to take the Petri dishes themselves, and those become like smaller sculptural objects or cultural artifacts, I like to think of. And I'm able to play with creating patterns within patterns. So in this particular installation, if you move far back enough, within the dot matrix of the Petri dishes, there is a larger pattern that exists. So I also like playing with this like push-pull relationship between the viewer's body and scale. And creating tension between like small scale and large scale and so on. And I did have a video in here, but it's not working for some reason, or maybe this is it. I don't know if you can see it. But again, you can see here the image as it's moving. The source material is what you saw in the very beginning. The close-ups of the Petri dishes, the growing, decaying, rotting food. So everything that you see, everything that I've shown so far, the source material is Maluba, believe it or not. And I've been collaborating with my grandma. Hopefully she'll stick around longer, and sort of collaborating with her to collect recipes, thinking about cultural legacy through food, community, family, and so on. And trying to extract as much information as I can while I have her around. So that's been really interesting for my work. And I'll just move to the next slide. These are more recent projects that I'm playing with, where I'm thinking about patterns in domestic spaces, but also moving between a scientific space versus a domestic space. So here, again, being more meticulous with how I'm playing with the leftovers and thinking about the materiality of the object of the Petri dish. I've been drying out some of these ingredients in agar and kind of creating these, like, I think of them as like agar chips or like agar leather. It's really weird and it smells really bad, but it's really cool. And I feel like I'm onto something with this. I don't know what just yet, but stay tuned. And so I'm also able to take some of these images here and just playing around with manipulating different colors. I've been printing these on wallpaper, and I'm working on an installation where it'll just be like a huge wallpaper installation where you're engulfed by these patterns, colors, and Petri dish prints. So you can see here some different colorways that are inspired by a number of different things. Also, I think I'm running out of time, so I should probably wrap this up. And then some other things that I'm working on is thinking about the failures of translation. Again, when you're moving from one place to another, in this particular case, moving from Arabic language to English language, thinking about the nuances of language, failures of translation. So in this particular piece, Gharba, the sign flashes on and off between one letter, and the variants, both words are derived from to be estranged or to be a stranger. Gharb meaning west, and Gharba is how you describe someone who's moved out of the Middle East anywhere west or to Europe. And so the variance of one letter to move west is to be estranged. So in this particular work, I'm thinking about this sort of like cautionary tale in a way. that it can be inviting, but also a very solitary experience. I've also been learning how to bend neon, which is really difficult. I might lose an eye in the process, we'll see. And so this is a very new piece that I just made earlier this year that reads Kanya Makan, which is the Arabic opening statement or inflection of, it translates to like once upon a time, but the way, the nuance of Arabic language, it's also speaking to once upon a place. So again, thinking about nostalgia, placemaking, and home, and this duality of existing and belonging to more than one place. So you can see just conceptually the things that I'm very interested in exploring, and how I like to move through food and through a number of different material objects to discuss those things. I'll just leave it there for now because I want to leave room for other people. But I will say I didn't get to talk about matluba very much, but if anyone wants the recipe, please email me and I can send it to you. I'd be happy to share. Thank you.
Thank you, Jude. Maybe you can unshare your screen now. Yeah, okay, thank you so much. This was a very interesting and visually compelling presentation. So now I think we have Laura Markley. And hi, Laura. You're welcome.
Oh, can you hear me?
Yes, yeah, we can hear you.
Okay, cool. 1 second.
And we can see the screen. At least I can see the screen.
Okay, awesome. So I don't have a timer with me, so just let me know if I talk too much.
Okay.
So, sorry, I'm moving around the little faces to make sure they don't block the screen. Okay, so I'm a PhD candidate in civil and environmental engineering, and I'm really interested in the life cycle of plastic and different aspects of that and different impacts of plastic on human and environmental health. So one of the studies I'm working on is microplastic pollution. So it's more the end of life impact of plastic and how it can have these long-standing impacts in the form of microplastic, and specifically looking at some regional occurrences of microplastic. So first I wanted to start out with something that's like a little interactive and fun. So this is actually a sample I was just looking at the other day, and I just thought it was really cool looking. So just take a second to see if you can see any potential microplastic. And I'll kind of point out where I would look personally, but okay. So there's kind of a giant bug in the way. So that can definitely impact where you look. But I would look at all of these for just an example. So there's this, I don't know if you can see my mouse, but there's this blue piece on the right that I would probably look at. There's this long fiber that's here, another fiber here, and then there's a fiber on the bug body. And there's also a couple of pieces here that are harder to see, but they look like pieces of plastic film. So I would also probably look at those to see if that's a bug carapace or an actual plastic film, which you can tell looking further up close. But I thought that would just be a fun exercise to look at plastics, and especially how similar they look to a lot of natural things, which makes it very difficult but fun at the same time. So to do a general overview of the topic, so plastic production is increasing from the start of plastic production in around the 1950s to now. And most of plastic that is produced for packaging, where it's used for the least amount of time. And that's where a lot of the waste comes from. So mismanaged plastic waste. And most of it never gets recycled or incinerated. So it can end up just sitting in a landfill. It can be sent to another country, which is often what happens, and it pollutes land in various degrees. So plastics can be released into the environment as microplastics, which are 1 micron to 5 millimeters. So I measured this earlier, and for my own fingernails, that's about 5 millimeters is half of a fingernail. So they're not tiny, tiny, but they can become really, really small as they break into smaller pieces. And they're kind of categorized into these different categories. Primary microplastics originate as this size range. So if you think of microbeads in personal care products are supposed to be really small. Secondary microplastics are made from the degradation of bigger plastics. So if you think like a plastic bottle that gets into the environment can eventually degrade into smaller pieces of plastic. And then microfibers, which are kind of within both categories. So those are released from washing clothing. They can also be released just in the air as you wear clothing, as you walk on a rug. You're probably releasing tons of microfibers, not to like terrify you, but they're kind of everywhere. So we're living on kind of this plastic planet full of microplastics. And microplastics can actually act as vectors of contaminants. So not all plastics are the same. Chelsea Rockman, who made this figure, has a great statement that microplastics are not microplastics because they can vary so much. So if you think of all the different plastic types that you see when you're recycling, those are all made of different chemical ingredients and different monomers, which are what link together to form the polymer. So if you think of something like polyvinyl chloride will contain phthalates. Polycarbonate plastics will contain bisphenol A or BPA, which I'm sure a lot of people have heard about. And polystyrenes can contain styrenes. And all three of these are chemical ingredients, whether it's a monomer or an additive, that are potential endocrine disruptors or carcinogens, which can have negative impacts on humans and aquatic species. There's also some chemical byproducts that can form on plastics, and plastics in the marine environment become an ideal surface for different contaminants to sorb onto and be transported. And they can even transport different bacteria, and that's a whole nother area, but it has some interesting implications for health, which are still an area of continued research. So in marine invertebrates, ingestion of microplastics has been linked to reduced reproduction, growth, and fitness, whereas in humans, ingestion or inhalation has been linked to inflammation, but there's a lot of unknown effects because this is still a very undeveloped area, so a lot to continue researching. And there's a lot of different potential sources of microplastics that could get into a lake. So this is just looking at some of them, but you can get things like degraded litter from stormwater, combined sewer overflows, personal care products, which can release microbeads. Wastewater treatment plants can also discharge effluent, which may contain microplastics. Soil amendments can also contain plastic, especially if they are sourced from sewage sludge from wastewater treatment plants. Boat coatings are also designed to slough off of boats, so those often can contain polymers. Snow is also another source. Car tire fragments from abrasion against asphalt, and industrial additions as well, so plastic resin pellets. And microfibers can originate from laundry as well as air. And microplastic abundance in central New York is poorly characterized, and freshwater in general hasn't been looked at as much as marine water. And this is just showing a map from Adventure Scientists from their 2020 map, showing just where the plastics per liter and where studies have been done. And where I'm looking, there's not a lot. There's a little bit in Ithaca. For previous work in this locality has been the Laurentian Great Lakes. So Erickson et al. In 2013 did a study on Lake Erie, Huron, and Superior, and they found a pretty high abundance with an average of 43,157 plastic particles per square kilometer. and the highest being 450,000, which is crazy. I don't think I'll find that much, but it is very interesting. And Onondaga Lake eventually feeds into Lake Ontario. So I thought it would be interesting to look at that lake. So no studies that I am aware of have looked at microplastics in Onondaga or Skaneateles lakes. I'm sure most of you are aware of both of the lakes. Onondaga Lake has a lot of historical pollution, primarily mercury, and it's been remediated a lot, but it still receives up to 20% of its inflows from the Metro wastewater treatment plant, which is a potential source of microplastics, as well as being in a highly developed area, so a lot of potential litter inflows and some combined sewer overflow inputs as well. Whereas Skaneateles Lake is kind of our comparison or analog because it's assumed to be a highly pristine lake. And I was also interested in this lake because it's the unfiltered drinking water source for the city of Syracuse. So any microplastics that are there potentially could end up being ingested. So just showing a map of our study location. So you can kind of see all of this red area is developed, and this is around Onondaga Lake, whereas Skaneateles Lake has mostly agriculture, forest, as well as a small developed residential area. But I'm going to be looking at the abundance, spatial and temporal distribution, potential sources and fate of microplastics, primarily focusing on Onondaga and looking at the inflows, so Onondaga Creek, 9 Mile Creek, as well as the center of the lake with nets and grab samples, and Seneca River and Oswego River. And then doing some net tows in Skaneateles as well. And so this is just to show what the net looks like in action. So it kind of has these two flotations on it, and it floats and collects from the surface water. And then this is another sampling protocol. So this is my trusty bucket and natural fiber rope. So I end up taking this into the field, and we have another Home Depot bucket. We sieve through a 335 micron sieve into the other bucket and kind of collect that to see how much volume we're sampling. But we get around, I think 37 liter samples that way with some volume reduction. And to kind of summarize this, because I don't want to go through the whole methods because it's boring, but samples are processed to remove organic matter, so things like leaves and detritus, which could be mistaken for microplastic or kind of cover up microplastic. And we do that with wet peroxide oxidation, but kind of looks like this. I call this the sample sauna. They kind of go to the spa and have a day and hopefully come out a lot cleaner so we can look at them afterward. And then samples can be vacuum filtered on the gridded filters for visual identification. And this is kind of what that looks like. So we take out all of the water to make it easier to look at the samples, and we put them on this gridded filter, kind of like what you saw earlier. And then we're able to count the plastic based on a set of criteria. So each particle is described based on its color and morphology. And usually if I have like a 50% certainty that I think it's plastic, I'll pick it out, and we'll later try to identify those in Raman spectroscopy. And so some of the different categories, they're mostly based on pressure and give, and they're not clearly stating what we think the source of the plastic is, but more describing the shape and morphology and the overall feel of the plastic as well as its color. So just to give some examples from a recent sample, I thought it would be fun to look at some close-up pictures of what some of these look like. And just to give a disclaimer that none of these have been done through the Raman, so they're not confirmed, but these are highly likely to be plastic. So one example of degraded litter is this piece here, which is actually a macroplastic, so it's bigger than 5 millimeters, as well as this piece of balloon taken from the inner harbor of Onondaga Creek. Another potential source is personal care products, which I actually found some potential microbeads the other day, which was exciting and also sad, but they kind of look like tiny beads that are colorful. It's things that used to and shouldn't still be added to face exfoliants and things like that. Another potential source, industrial plastic resin pellets. So this is an image from Wired on the right, and then my image on the left. So there are spheres with these small indentations in the middle. And then another source, which is the most highly reported source, is microfibers. And they just look like small fibers. They tend to have no tapering toward the end to distinguish them from natural fibers. So what's next? I'm still finishing processing and counting samples, and I'm going to analyze our microplastics after picking all of them to determine that they are plastic and also what type that they are. And we're going to continue sampling during different seasons to see if there's any seasonal changes in microplastic with increased flow conditions and things like that. And we're going to also vary the volumes of water to see if it has an impact on plastic counts. I just want to acknowledge all of the sources of funding. The USGS is funding this study, but I'm personally funded through the NSF, Empower, and my department. And I'm doing a lot of this work with the Biomaterials Institute and Earth Sciences, as well as my undergrad, who has done a lot of great work with me, Eleanor Hugh. She's been awesome.
Thank you, Laura.
So now we're going to go into a 5-minute break. Thank you to Jude and Laura. Those were some great first talks. I enjoyed both of them. So take a break, go to the bathroom, come back. We're going to finish off with some awesome talks afterwards.
Yeah, I just wanted to say that we are coming back in 5 minutes, which means 6:50. Yeah. Okay. See you in a bit. I hope we are all back. And we can continue with the next presentation, which will be Daria Warner. For those who missed the introduction in the beginning, she is an MFA candidate or already graduated from the University at Buffalo. And she's an artist. So this is from the art department. And I'm actually really curious to hear what she will be talking about. Daria?
Thank you, Brianna. I wanted, let me just let me know if you can hear me, right?
Yes.
Okay, great. And now I'm going to share my screen. 1 second. Okay. So just let me know if you can see me and hear me, right?
Yeah, you can. I can hear you and see you, and everything I think looks fine.
Okay, perfect. So first of all, I want to thank you, Brianna, and thank Heidi for inviting me to this BioArt Mixer. I'm super excited to present this project. I've been working for the past 3 years. And I did graduate, so officially done with school. And so this project literally took like 3 years to finish, and it's still, I'm working on a new prototype again, but overall, basically, wait, I'm just gonna hide this. Yeah, so before I go into details about this project, I want to address some of the background and why this whole thing started. So I was born in the Ukraine, and in 1986, Chernobyl disaster happened. And on that day, my family was supposed to fly to Kiev, which is right here, as you can see, and it's probably 90 kilometers from Chernobyl. So obviously everything was canceled. But for the next 5+ years, every time I visit my grandparents, I wasn't allowed to touch anything outside. I wasn't allowed to play with anything outside. We had to go home, wash our clothes, wash the shoes, close the windows. And it was pretty devastating because as a child, I was used to playing outside and touching everything. And then my family got relocated to Belarus, and and then it was just this anxiety constantly of like going to the market, where's the food is coming from, right? Is it contaminated? Because any food that's 70% of Belarus was contaminated with radioactive fallout. And if the food was coming from the areas where it's coming from, the only way to know was to have a Geiger counter, and there was some kind of governmental control, but again, I don't think it was very clear. So, and my family decided to stop foraging for all the mushrooms and the berries from the woods because we wouldn't know if they had accumulated toxins. And I want to brush on about what the mushroom cycle is and why the mushrooms will accumulate the toxins in the first place, which brings us to the fungi life cycle. So when we go to the woods, collect the mushrooms, we collect in this, right? So this is called the fruit body. And it just appears on the top, right? So we're all familiar with that. But what's invisible out there is the mycelium, which is right here in the right corner. And it's a vast underground network of cells combined together. It's called hyphae, and it spreads all over. It holds our soil together. It decomposes and creates nutrients and extracts nutrients from the soil as well. And when mycelium sense change in the environment, either humidity or temperature, it produces these mushrooms as a response to the stress, which will then produce spores that will be carried away in vast territories, so the genetic material will be spread. So mushrooms, so basically mycelium extracts the nutrients from the soil or substrate it's growing on. So in this case, it pours all this energy into the mushrooms, which naturally makes it, accumulates anything that's in the soil in the mushrooms. So this is the reason why mushrooms are the ones like who are great decomposers. They can biodegrade toxins, and oyster mushrooms are really famous for that. So this was so again, I wasn't able to forage for new mushrooms, and kind of like this mushroom phase of my life was put on hold until I came to School of Visual Arts, and I took an undergrad class, undergrad program with Suzanne Anker and her mentorship in bioart. And one of the workshops that was presented that completely blew my mind was the mycelium workshop, where we would grow living sculptures out of mycelium. So since mycelium has this unique ability to take over the substrate, which is basically the food it's digesting, I was blown away and realized that, first of all, I can grow mushrooms. Second of all, I can actually know where my ingredients are coming from, so I can control the outcome. And third of all, I can create a living sculpture that could continue growing beyond my control, which I was very interested in. But one part of this project, or working with this material in a sculptural way, was you need to create a mold to do a sculpture, right? So in this case, plastic, unfortunately, was the best option because it was non-porous, and you need a non-porous material for mycelium to grow in, so to avoid contamination. So for me, this was like a huge disconnect, because I was like, well, on one hand, I have this amazing organism that can degrade the toxin and pollutants in the soil and transform. And on the other hand, I'm using plastic and producing waste. So this is when I kind of started thinking about sustainability in art practices, and how we as artists address this in progress environment that we create, and what happens with all the pieces that never make to the show, and where is the balance and how can we find it. So this is how idea of the microprinter was born. And microprinter is an open source 3D bioprinter that prints mycelium substrate ready for inoculation with various types of fungi, and it combines additive manufacturing with biological tissue to create a living sculpture. So in this summer of 2017, I was looking for a scientist to collaborate on this project, and I met Puneet Kishore, who was a data analyst and programmer and engineer, and we wrote a proposal together, sent it out to several residencies to build this prototype. We got into Korg International Artists Association Art and Science residency for a month in Delhi, and we went there and built this very first prototype right here. It's based on Tiva Tarantula, which was a self-assembly 3D printing kit. But the main part of this was this syringe pump that was donated by our third collaborator to us, by Neves, who was based in Madrid and was finishing her PhD in creating a 3D bioprinter to print skin tissue for burn victims. So while Puneet was deconstructing and reassembling the printer again, so it was able to use the syringe pump, I was working on substrate research. And since we didn't have access to the lab, we set up this really like very low key lab in the kitchen, and we spent a lot of time in the kitchen. And part of like Indian culture is cooking. So I was like, how can I connect the local space, this local site, to the actual project that we're working on? So I decided to experiment and introduce mycelium to different substrates. And so starting from the left here, I have rice flour and red pepper, coconut fiber, rice flour, black pepper, vegetable agar, coffee grounds, turmeric, chutney, and the last two, I don't even remember, I think it was a combination of different things. Since we didn't have any access to any lab equipment to do analysis, but we were able to actually see, just like visually observe the results. As you can see, chutney here doesn't have any growth because I think there was a lot of salt present. And then mycelium really enjoyed coffee grounds, and also rice flour was very, responded very well to it. Turmeric, however, was, having trouble. Mycelium was having trouble growing it, and I wonder if it's because it has antiviral, antifungal properties in some, alternative medicine. And the end of the residency, we had a show, and we had an opening for one day where we 3D printed our first sculpture, which was actually not kind of like sculpture, but more of the two-dimensional. It was like 3 layers, and we 3D printed a symbol of Om, which is in Indian cultures like Hinduism, Buddhism represents, it has like this essential meaning of either everything or the soul and the essence. And this was in a way a notion or metaphor for kind of like all the living matter has a soul or an essence, and like how can we address that? So following that, when I enrolled into university at Buffalo in 2018 in the fall, so my 2018-2019 year was filled with substrate research at Coalesce Biological Lab. So I realized I ran into several issues which I need to figure out. Like I need to create a gel-like substance that will be printable but hold the form. It has to be porous so mycelium will integrate. Gotta be local, if not, but over the counter, something simple, ingredients, non-toxic, something that accessible, because the whole point of this printer is to be low tech, is to be completely, it will be inexpensive to build. So here I experimented with tapioca flour, rice flour, and cornstarch to create this prototype of the substrate, understand the properties. I was consulting with Dr. Ann Mayer from Biomedical Sciences Research on biomaterials that really helped me a lot. So mycelium did respond well to this, and so gave me a lot of good ideas on how to pursue this further. And my next semester, my final year here in Coalesce, I focused on my thesis where I actually started building the actual microprinter, the second version. So I did research and realized that the best option out there to base my printer on was Delta 3D printer with clay extruder. And I found open source designs provided by Jonathan Keep, incredible ceramic artisan / engineer. So they were open source. So I built this printer from scratch. And since I don't have a lot of engineering background and very little coding, so this was quite a ride also because I had to figure out how to calibrate this printer according to specific substrate that I'm creating. So, and it was like trial and error, like coding involved, coding doesn't work, things fall apart, like, spend like 2 weeks figuring things out, like one little thing, and then suddenly it's all working. And here you see printer working, and the very first sculpture was like this, obviously, wasn't working precisely. So after calibration, it was meant to print in something like this. So I did some rice flour experiments and other substrate that I have been working on. And so my thesis was supposed to open on, April 30th. But obviously because of the pandemic, everything is postponed. And while I was working on this printer and I finished it, I was thinking how this printer it's a travel article. Basically, it's a part of the big series that I'm now researching to explore. It's a site-specific installations that will be based, it will be addressing local ecologies. And the first site I wanted to address was Buffalo China Factory, located here in Buffalo. And I had the factory was over 100, it was operational for over 100 years. And it was at some point, I think in the beginning of the 20th century, it was the only factory in the world that was completely running on electricity. And I had an opportunity to go and get a final sale of some of the china in 2018. And I had a tour of the factory, and it blew my mind. It's like it was filled with this shelves and shelves of these bisque plates. They are soft plates made of kaolin clay, and they are ready to be fired for kiln, but they will never be there anymore because this factory is not operational anymore. So these plates were just sitting out there, and this whole factory looked like this giant leviathan that just fell asleep after all these 100 years of operation like this. And I was wondering, is there a way to give this place, to give this space, to give this entity a new hybrid life in a sense? So what I did is I used one of the cups that I purchased at Buffalo China Factory, which as you can see right here, it's a green cup. And I 3D scanned it with the software at school, and I had to clean it. So my software of my printer was able to understand and 3D print it with my substrate. And then I created a very specific substrate, which was based with the bisque plates, which is kaolin clay, with the coffee grounds I collected from the local Starbucks, and these shavings from the cardboard boxes where I got my deliveries when they were sending parts for the printer to be built. And so it's very important that the ingredients, again, I just want to emphasize this, either local or, not sure why my video is loading really slowly. Sorry about that. The ingredients are either local or they are, you know, are partially local, or they in some way address the locality of the site, because it's important to offset the carbon footprint for me. So this whole printer is built under $1,000. It's about $800 for the whole thing. So it was also important. And I use like scrap wood and as much to offset again on my carbon footprint. So one of the things that happened along the way, which I did not anticipate it, but one of these happy accidents happened in art. 3D printer is all about precision. And yes, the printer calibrated to certain precision. But sometimes, since it's an open source and the code is not perfect, it will encounter a glitch. And the printer will either stop printing or made a mistake. And these mistakes were absolutely beautiful. I thought they were more precious to me than the actual cups that I 3D printed later. I thought they were absolutely beautiful, amazing, and it was so just one of the kind, because the printer will never be able to repeat this. They were just there manifesting themselves, and I thought it was like an interesting thing. Like kind of like a collaborative. I know it's just a glitch of a printer, but for me it was like a collaborative artwork with the printer itself. And so I printed 4 cups, seeded them with 4 different types of mycelium oyster mushrooms. One of them is a wild strain collected here on site, and in about 11 days into the seeding, you can see mycelium overtaking, responding well to the substrate. The process is working, mycelium like this substrate, and it's growing and overtaking these cups. So basically this, about 30 days later, the mycelium produced the actual fruit bodies. And here you see the beginning of the fruiting bodies, right? And here you see like the full mycelium, the actual pink oyster mushroom coming out. And all these little tiny dots or little branches you can see right here, they actually are future fruit bodies. So the idea for the installation would be to display 6 pedestals in a Buffalo China Factory with people, with audience coming in the beginning of the show to observe the first stages of this fruit body growth. And then since the mycelium, when it produces the mushrooms, it will pull all the energy out. So the cups will start degrade and deform. And so the audience would be encouraged to come back in 2 weeks to see how the mycelium will shape the sculpture itself. And I wanted to also emphasize the idea of like, you know, chaos and control. What is chaos or what is control? Like factory, industrialization, and how we are so like mm, fixed on everything being precise and perfect. And this is, I don't want this to be not precise and not perfect. And I think the beauty, and nature doesn't work like that. Nature works in aberrations and irregularities. And that's why I think nature is really beautiful, because all of that. So the second part, the final part of the show would be that once mycelium produces these food bodies and release the spores, and they will be collected by the air currents of the factory. And the factory is actually pretty dark, and because it's not operational and it's very damp. And the spores will seed themselves into the factory and produce another vast networks throughout, and basically maybe creating this new hybrid entity between the factory and the mushroom, and giving like a new kind of life. So I wanted, with my thesis, I wanted to address like the ideas of what's living versus non-living, maybe the idea of like possibly intermatter and intermatter communications, and how, like the ideas of control, and also as well as sustainable art practices as well. So microprinter workflow could be found on my website. It's just a little schematics that I created to show, like, I'm working on a couple of recipes, and right now, and so basically there's different applications. There's art and design, there's just art. And right now, the broad applications and the future plans, I'm waiting for a response from from collaboration with the Greenhouse in Colorado next year, to actually create, again, the site-specific living sculptures, but collaborate with the architectural context, with the production and growth of custom-made planters that will be inoculated with mycorrhizal fungi to promote better growth and yield of starter cultures. There's also a design, obviously, application for that, the production and growth of custom-made biodegradable products and packaging in combination with 3D scanning app. There's companies already have been doing that, and on a massive industrial scale, like Ecovative and some other companies like MecoWorks is working on that too. So the educational aspect is really important as well. It's important to engage local communities through sustainable art practice workshops, because I think as an artist, we sometimes possibly forget that we think the message is very important, and the process and what we create within the process while we deliver this message sometimes becomes irrelevant. And then I've seen a lot of trash coming from the art departments, and that just really was discouraging to me at some point. So I think that there's a balance there that we can achieve. And if we talk more about this, and in a very humble and sustainable way, there's a way to find this balance and possibly teach future generations to address these issues in a more eco-conscious way. So I'm also open to disciplinary practices and collaborations in STEM and design, which brings me to my final slide, where you can see my contact if you want to reach out, or you have any questions, or you want to maybe correct me with something that I said something, or you want to advise me on something. So this is my credentials, like the email and my Facebook account and Instagram. And I want to thank again Mariana and Heidi and everybody involved in this, organizing this BioArt Mixer, especially in connecting people and sharing these ideas. Thank you very much. I'm just gonna stop sharing. Okay.
Thank you, Daria.
That was awesome. Next we have Juliet, and she's in Susan Park's lab, trying to get her PhD. And she's in the biology department here at Syracuse University. And I'm pretty excited. She looks at marine animals and how they communicate and the sounds they make. So.
Okay, let's see. Do you see my screen?
Yes. Yeah, we can see it.
Okay, great. So yeah, today I want to tell you guys a little bit about part of my dissertation research, focusing on the vocal development of humpback whales in Hawaii. So this is a picture of some of the humpback whales that I got a chance to see in Hawaii this past year doing some field work. So the first thing that I want to do is show you guys a little bit of humpback whale song. So switching gears here and doing a little bit of audio. Let me know if there end up being any issues with the audio if you can't hear it. But first off, just to orient you to what you're looking at, this is a waveform on the top and a spectrogram on the bottom. So both of these are visual representations of sound. On the top, we have the relative loudness of the sound over time. And then on the bottom, this is showing the frequency or the pitch of the sound over time. And then how loud the sound is shown by how dark the color is. And so there's a green bar here that's going to move across the spectrogram so you can see what you're listening to as well. So this is an example of just one small part of one humpback whale's song, and I'm going to revisit this again later in the talk, but I want to first orient you a little bit to what animal communication is and how animals use sound. So a lot of different variety of animals use sound to communicate. So birds use sound to communicate, mammals like whales use sound, insects like crickets, fish, frogs, so a really wide diversity, and they use sound for a lot of different things. So animals might use sound in order to find a mate, in order to, for social cohesion within groups, in order to tell others warn about predators, and a variety of other contexts. And these sounds can take the form of very simple, singular sounds on their own, and they can also combine to form sequences of sounds. And so animals may use sequences of sounds in order to convey additional information through different characteristics of the sound, such as the timing in between different notes, the differences in the notes in terms of their pitch, potentially whether there's rhythm in the sound. And even among these different sequences of sounds that animals might produce, there's a lot of variation in how complex they are as well. So here you can see some just cartoons of what that might look like. And so even though different animals might all produce sequences where there's repetition, where there's multiple sounds together, strings of sounds, there might be more complexity in terms of how many different types of sounds are used, or how often they're repeated, or different things like that. And so I'm particularly interested in how animals are using these more complex acoustic sequences. And in particular, song is a type of acoustic sequence that a number of different animals use. And so you might be most familiar with bird song as an example of animal song. So birds use song in a variety of different contexts as well, including attracting a mate and defending a territory. And a lot of what we know about song comes from birds. And we know a lot less about song when it comes to mammals like whales. And so I'm really interested in song because it's this complex form of acoustic sequence that has really interesting elements of rhythm, of patterns, of different types of notes being involved. But the question of how we define song is a really complicated one, and is something that I'm trying to grapple with a little bit with my thesis, because there's what we think of as song as humans and in human culture, what song and what music are and what that means to different people. And then there's also what it means for different animals. There's differences in the structure of the song, and also in the function, in what these different animals are using it for, whether it's used as a performance display, whether that display is aimed at attracting a mate, or at fighting or defending a territory against others within the same species. So there's a lot of variation here, and trying to figure out what song is from an objective standpoint is something really difficult, and something that is a really complicated part of the research that I'm doing. But one of the other reasons that I'm so interested in song is because oftentimes it's learned by the animals. And so this involves an even more advanced cognitive ability. And so, for example, most bird songs are learned within the passerine birds, the songbirds. And in a lot of cases, these animals listen to other animals producing those songs and then learn how to imitate those. And that's how they learn to produce their songs, as opposed to innate production of song, where they don't need to listen and learn how to produce that song. They can just do it immediately right away. And so a lot of research has been done on the song learning in birds, in particular because of the potential parallels to human language development is one of the reasons that there's been a lot of research in this area. And so we know that birds have this initial phase where they're listening to other birds around them, producing songs, and then they produce this plastic song or sub-song, which is when they're trying to produce the song, but it's not perfect or it's not very good yet. And then eventually the song crystallizes and they have the song that they're going to sing for the rest of their lives. And so this can be compared to humans who also spend time listening and then spend time producing, spend time babbling, which is similar to the sub-song or plastic song that birds produce. So it's this practice at making sounds. After listening to the adults make sounds, it's practice figuring out how to use the actual motor mechanisms that are used in order to produce sounds. And so I'm really interested in figuring out more about this, these different learning pathways, particularly for other mammal species, since we don't know as much about how song or other vocalizations develop in some other mammals. And specifically, my study system is the humpback whales, and we know that humpback whales are able to learn their songs because humpback whale song is culturally transmitted. So humpback whale song is the same across all individuals in a given population. So there are multiple populations of humpback whales all around the world. And within those populations, all of the whales sing roughly the same song, but that song changes from year to year. So it's been compared to human pop songs, because new songs will arise and then we're able to watch as those songs move across different populations and take hold in new populations until everyone is singing them. And then within the next year, it moves on to the next popular song. And so because these songs are changing from year to year, we know that the whales must be listening to each other and learning how to produce the new song that way. And so I'm using this study system because we know that it's learned already, but I want to know a little bit more about how that learning is taking place in animals from a young age. And I just want to give you a little bit more background on humpback whales. Only the males sing among humpback whales, or at least only males have been recorded singing. There's never been a female recorded singing. And song is predominantly produced on the breeding grounds, although it is also recorded on migration and on the feeding grounds. Humpback whales undertake migrations, you can see on this map here in the North Pacific, each year between feeding grounds at higher latitudes and breeding grounds at lower latitudes. And it's because of the fact that song is mostly found on the breeding grounds that we think that song is associated with breeding somehow. But at this point, we actually still don't know what exactly the function of humpback whale song is, despite decades of research on it. Because it's not clear that it attracts females to males or helps in mating in any particular way. And so, as I was starting to say before, past studies of song and other humpback whale vocalizations have focused mostly on mature adults. So that's how we know about the cultural transmission of song and how they're learning from each other. But what we don't know is what some of these sexually immature whales sound like. So there hasn't been a lot of research on younger individuals and essentially what I'm interested in looking at is what some of those teenage adolescent whales sound like if they sing songs poorly, if they're practicing those songs, if they produce songs at all, or if it's like a switch and suddenly turns on once they become sexually mature. And so in order to do this, I am really lucky to be able to do my research in Hawaii. So this is a picture of Maui from the water, from the boat. I got a chance to go this March to Hawaii. And I'm doing my work in the Hawaiian Islands Humpback Whale National Marine Sanctuary. So part of the sanctuary is located right off of Maui. And so there's a little house icon here showing where I was stationed. And right in between the islands of Maui and Lanai and Kaho'olawe is where I go out on the boat with the Marine Sanctuary team, and we would tag whales in order to study what sounds they're making. And so, like I said before, my goal is essentially to try and figure out a little bit more, just start to characterize what this vocal development process for humpback whales look like, because we have some idea of what that looks like for birds and also for humans. And it would be really interesting to find out if humpback whales are following a similar process in how they're learning, or if maybe their learning process does look something more like those animals which have innate song. And so in order to characterize some of what this development is like, I'm looking at a bunch of different age and sex classes on the breeding grounds in Hawaii. And so in Hawaii, we have a lot of different whales and different behavioral roles. So we have moms with calves, like the one you can see here. This calf was probably only a few weeks old at most. And sometimes these moms with calves are also accompanied by adult males. And then we also have competitive pods where there's large groups of males competing with each other, potentially over access to a female, but we also know nobody has actually seen humpback whales mating. So we actually don't know when or where that happens exactly, but we know that we do know that breeding is going on these breeding grounds. And then some of the other roles that we see are individual singers, which are usually on their own and are down underwater at depth, singing for extended periods of time. And then there's also occasionally dyads, groups of a male and a female swimming together. And in order to study some of these different behavioral roles, these different ages and sexes of animals on the breeding grounds, I'm using acoustic tags in order to record the animals' behaviors. So on the left, you can see an example of one of these suction cup acoustic recording tags. This one is called a D-tag, or digital acoustic recording tag. And they're attached with suction cups to the back of the whale, and they record the movement of the whale. So they have accelerometers and magnetometers in order to figure out how the whale is moving in the water. And then they also have hydrophones, which are underwater microphones, in order to record the sounds that the whales are making. And the way these tags are attached is by a long carbon fiber pole from a small boat. It's stuck to the back of a whale. And then these tags stay on anywhere from a couple of minutes, depending on the placement and the whale's behavior, up to, we've had tag deployments up to 48 hours. And so I just want to show you a quick video of what that actually looks like to deploy a tag on the whale. It looks a lot easier in this video than it actually is. But this is, we got the boat into a competitive pod of humpback whales. So you'll see a bunch of different whales, and then the boat pulls up alongside the whale before the tag is deployed on the back of the whale as it surfaces right alongside the boat. And so it's really cool also about, especially deploying the tags in a competitive pod, is these whales are so busy paying attention to each other and fighting with each other that they don't even notice the boat or the tag being deployed at all. There's absolutely no reaction to the boat and to the tag. They just keep on going. And so the other really difficult part after getting the tag actually on the whale is that we also have to go collect those tags in order to get the data, because we have to bring the tags back on land and plug them into a computer and download all of the data. And so in order to do that, we use a radio tracking system. The tags make a beeping noise. They float to the surface and make a beeping noise, and then we have to follow the beeps, kind of like a scavenger hunt to find the really expensive equipment in the middle of the ocean. So here's a picture of me in Hawaii with some whales in the background, using the antenna that you can see here and listening closely for beeps to try and find our tags. And then once we download the data from the tags, that's when I get to spend many, many hours listening to sounds like this. And I'm going to play just a little bit of this again to remind you what they sound like. And so I also want you to notice here what you can see in terms of the pattern and rhythm of the song, because you'll notice that some of these notes are repeated, but there's a little bit of change in some of the notes. And so this is something that, having just gotten these data this year, I'm starting to really dive into is what are these different units, how are they arranged, and how does that differ between individuals, especially over those different age and sex classes. Because by looking at the differences between individuals, if they're of different ages, I can also get some information about, okay, are the ones who are younger singing significantly differently from the mature adults? Are the mature adults all singing the same kind of song and the juveniles singing some crazy different song or a bad version of that song? And so this is something that I'm diving into right now, and I'm really excited to see what the results are going to be. And so then I just wanted to briefly mention why any of this matters, why we care about how humpback whale song develops. And so the other part of my dissertation is that I'm framing this in terms of the evolution of song in mammals as a whole. Because by looking at how song develops, whether song is learned and how song is learned in whales, and then comparing that to what we know about other species, we can learn a lot about evolutionary trends. And that also has the potential to tell us something about human music and language and how human sounds are learned and produced and how they evolved. And the other thing is that by understanding whale behavior, we can help in conservation efforts too. So there are these really complicated vocal cultures that different whale species have. Humpback whales, but also sperm whales, killer whales, and dolphins as well, where a lot of cultural knowledge is passed between individuals. These animals can live upwards of 100 years, and so there's a lot of cultural memory embedded in these different sounds. And so by understanding what those sounds are and what role they play, we understand how important they are to the entire network as a whole also, and what are the fundamental units that need conserving and which need protection. And so I just want to end also with acknowledging the other grad students in my lab and my advisor, Dr. Susan Parks, and Dr. Mark Lammers, who I worked with in Hawaii, and my funding from the NDSCG Fellowship. And so I have one more picture of some whales, and this whale, it's a mom and calf pair and a tag, a bright red tag on the back of one of the whales in Maui. And then I think we can move into discussion.
Thank you, Julia. That was awesome. Beautiful. All the talks were really great, and thank you. So Tom is going to lead us into the discussion part of the last. So if you have any questions, write them into the participation. We can also help with Tom too. And
yeah, we can try to get some people up here. So thank you very much for those presentations. It's amazing. Congratulations to Borean and Heidi for using their networks to find these people and to put them together. I thought it was a very interesting pairing of people, putting people together. And I think that what I'd ask people to do is put your cameras on at least so we can see who the audience is. Because one of the most important things for the people presenting is to get a sense of who's been listening to them and to get a sense of what they thought, you know, which I think would be really important. I think I'd like to start the discussion in general terms across the four presentations. I found it very interesting that there were so many associations, and at one point we seemed to be coming from cooking into the Petri dish, and from the Petri dish into cooking. There were some strange transformations spatially going on. But I was also interested at the end when Julia was talking about these sound images, these acoustic images that form the patterns in language. I was very interested in, overall in the presentations of all of the presenters, because they formulated their own manner of pattern of these signals that would yield what they were doing in terms of their research, and allow us to get some understanding of the insights that they have into these collaborations with materials and with other life forms. I know that Daria has written quite a bit about the idea of working with, printing with fungi, dealing with kind of cooperative intelligences or patterns of intelligence when she works with other organisms. So my first question is, I'd like to hear the presenters talk about how do you form the messages that you form, the messages that we have? Are you in collaboration with the materials and the organisms that you're dealing with? And does that really, in a way, form a collaboration for you? I'm speaking loosely in terms of the environment, for how you communicate what it is that you're forming insights about. Because I think there were some very distinct patterns going on. And I'm curious if anybody can comment about that, the presenters, please. Unmute and let go. And
Daria here, so I don't know if anybody wanted to start, but collaboration is like a big thing in my work, and I've been very careful with that, because collaboration requires, I think, consent. And so how do we know if the organism working or not consenting to what we're doing, right? So I have been trying to kind of think about what are the ways, maybe like cooperation in a sense, or something. But for me, one of the most important aspects is to make sure that the organism I'm working with is having the best possible conditions, right? So I do the research about it, and then when I was growing mycelium, I was making sure, the best food, I did the research, what's the best food would need to be created. I feel like a lot of things are actually based on food, and the food makes us happy. So I was wondering what would make mycelium happy, right? What kind of food? So, and it's literally, I think right now what I'm doing the research, this project is just a part of the bigger research I'm working on different various systems of intelligence across species, and what could be the common ground where we can find, when we can talk between the species, right, and understand it, because it's almost, there's this notion of you can't be, in order to be a bat, you can't be a bat, you can understand what the bat is thinking. But I think there is a certain understanding, or at least there could be a certain kind of recognition to that, and into accepting and respecting these organisms' environments. So, and that's why it's important for me to address local sites and address local organisms, and to offset the carbon footprint, and make sure and ensure that the organisms I'm working with having the best possible conditions. It's I'm working with people in the lab. We want to make sure we're all in tune. So I just think I try to bring my work ethics into everything. So I think it's kind of what started me to think about sustainability in our practices in the first place, how, because I feel I've seen in a lot of artists, I think the message is so important to be delivered, which it is, and then everything that comes before it is just get secondary, which I think it's still important to think that way. So that's kind of my take on that. I wonder if I answered your question.
You did. I thought it was also, I just add that it was very interesting that you introduced this factor of noise or disorder, when you appreciated the hybridity of the responses of this material to the act of printing. I thought that was interesting. I thought that Laura, I didn't expect to see you being such a morphologist of the plastic fragments and particles, and I found that very interesting. When I looked at your website, it was more product analysis and an understanding ecologically of how things are harmful or where these microplastics come from. But I was very interested in your presentation that you turned to a certain physical morphology of the pieces, starting with that examination of the slide with the insect in the center and the fragmentation. But I just, in a way, I felt that you were collaborating or communicating with the materials that you were studying. Do you have any response to that?
Yeah, for sure. And I think that kind of lends to like the explanation of like where it came from, and kind of respecting the material in a way. Like a lot of people tell me with my work that plastic is bad, that we need to get rid of plastic, but I kind of try to approach it from the standpoint of something that's useful, but just being used really poorly. And I think looking at it that way and trying to treat every sample like that, instead of thinking, you know, I'm disappointed to see all of this here. Of course I am, that it shouldn't be in the environment. But it's also just interesting to approach it from, you know, where did these pieces come from and what is kind of their story, and what can I get from that by looking at like their morphology, their color. We're also hoping to do some other stuff with like isotope signature. And I just think it's interesting because you wouldn't think like a little piece of plastic could tell you so much about where it's been and where it came from. But once you get through it, it's just like an interesting story that it tells you. And yeah, I never would have thought my work would take me there, but it definitely has. And it gives me more, I think, respect for just resources in general, which I think sustainability is, you know, really all about. I thought it's the interesting part of, I didn't talk about this, but I'm working on some other stuff of just looking at humans' interaction with waste and what we think about waste, and trying to interact the human component of how we think of waste. And I've just always been really interested in how we perceive it.
Yeah, I thought it was so interesting when you look at the fragments, when you look at these plastic fragments microscopically almost, they almost have that kind of fractal kind of relationship with their, so we're used to seeing the milk cartons and all of these things, but I think it's very important to see that micro level as well.
Mm-hmm.
I thought it was really interesting when you started with the idea of having these dinners, having this exchange of food, and the communication that floats across that, and how important that is. But I thought it was so interesting that you were able to, you know, take that context and to take the food and to move it to the Petri dish and to these strange electronic signal patterns that you had with these kind of beautiful, almost crystalline, soft crystalline forms. And I thought that was a very interesting, like you were definitely communicating at another level that wasn't involved with speech and the transmission of culture over food in the normal way. So where do these forms come from? They were very consistent.
It's actually not as complicated as it looks, but a lot of my work is, I play a lot, and that's sort of how I discover and learn, and it's just, it goes hand in hand with just the way that my brain functions. I like to explore and I like to play, and part of that is bringing in like childhood ephemera, like a kaleidoscope, for instance. I feel like most of us have probably played with a kaleidoscope at one point in our childhood or lifetime. And that's something that's really ingrained in just how I perceive things. And so I've been playing a lot with mirroring, with reflections and refractions. So everything that you saw in my image making was just taking what you saw in the original source documentation of what was in the Petri dish and just splicing that image many times onto itself until it produced these very like geometric patterns. But there's really not much more to it than that. Like it looks, I'm talking it down, but it looks grander than what the process is when I come up with these patterns. But it's fairly simple in that sense.
Well, we know how a kaleidoscope works, how it intensifies colors and puts things into patterns, and we like that organization. But do you feel as though what you're doing could be evolved into some type of a code? I'm thinking about what Julia was talking about in terms of the repetition of acoustic signals into patterns which could be looked upon as songs, etc. But would you be interested in, or would it be likely that this kaleidoscope, this invigoration of the image would then turn into some type of pattern?
Absolutely. I think that at this moment, I'm still on the very like surface level of where else I can take this, how much farther I can push this process and where it can go from there. But even like I was having a quick side chat with Daria in thinking about the potentiality for the material itself being stretched through a printer using these traditional foods in that manner and coming up with like different sculptural objects. But even thinking about pattern making and what those sequences could then transform into, I think there's definitely something there that I'd love to explore.
And Julia, you left all these great things sort of hanging, like I had no idea that male humpback whales had not been recorded. I mean, that's completely new to me. And then other people were asking questions on the chat about why aren't the moms, what is the deal with these? What are the theories of the differences between sexes in whale acoustic signaling?
Yeah, there's a lot of different theories about what the function of humpback whale song is, and there's better support for some than others, but not really a clear winner. There have been, so a lot of the way in acoustic studies that we figure out what the function of a sound is by doing a playback. So by recording the sound of an animal and then playing it back to the animal and seeing how they respond. And so one of the theories is that humpback whale song sung by males is used to attract females. But researchers have done studies, specifically in my study site in Hawaii, where they've played the songs of humpback whales from that same year, and they have not observed female humpback whales approaching. And so it seems like there's no evidence for the song attracting females. It also, when we observe the lone male singers in the wild, there aren't females coming and assessing them, listening to their song, coming close. And so again, that it seems like it's not that they're attracting a mate that way. Some of the other theories are that the males are using it to kind of size each other up a little bit. So they're listening to what the different sounds are, what the different songs are that the other males are making. And then potentially when they come into those competitive pods later on, where they're fighting with each other, where it gets really physically aggressive, they're remembering, okay, I assessed this other guy singing earlier and he had a really good song, so maybe I don't want to engage and fight there versus someone whose song wasn't really good. And then they might not engage. There's other theories. There's a lekking hypothesis. So a lek in biology is when a group of males aggregate in one area and display together in order to attract females. So even though the individual males may not be attracting females, it's possible that since humpback whales are huge and sing so loudly and their song travels so far, everything takes place on a totally different scale for them than it does for us. And so it's possible that it's not that individual females are coming close to an individual male in a way that we would notice, but instead that maybe it's these larger populations, like the Maui population versus the ones on the big island of Hawaii, in that area, that maybe they're all singing together really loudly in a way that attracts females to that particular area within the larger Hawaiian Islands breeding ground. And those are just some of the main hypotheses. There's also ideas that maybe it's a form of sonar and they're using it to sense their environment, although there's not a lot of support for that one. And I think there might be a few others too, but those are some of the main ones.
It's fascinating. Okay. So the audience did not turn their cameras on for the most part. It's easier to see if people have questions if they go like, you know, let's do it. But are there any other questions from the audience? Because we have these people here and it's an opportunity for a discussion. I know Heidi had about 18 questions. And she's welcome to jump in too. Okay.
I think one of them, I don't know why I found it. Am I muted? No. Okay.
No, you're on.
I don't know why I was like so drawn in. Daria, your presentation, there was a couple of times there was like a little drawing of a chinchilla. I don't know what it was that popped up with your 3D printing. It was just like hanging out there once in a while. Is there meaning to that? Like, did you have?
Yeah. So it's the secret, my secret alter ego. So it's kind of ironic in a way because I started, it's a bat, right? But it was before the coronavirus. And when I mentioned the bat, I don't know if people have heard about it. Can you become a bat? There is this whole notion that you can't because, you know, you cannot have a bat consciousness. And even if you be transformed into the bat consciousness, you're still going to be a human consciousness. So since I've been working on this project and trying to like understand, you know, what's the collaboration and how you address it, the bat, I made this bat kind of like as a resistance towards the idea that no, you can't be a bat and you can't assess the conscious of the mushroom or a bat or anything else. So when I was building this printer, I just kind of like was having this bat next to the printer and making these videos, and I completely forgot that it's in the videos when I made my presentation. And so it's just being there, kind of like, you know, keeping me company basically. So it's like like constantly reminding me that maybe it is possible to become a bat or a mushroom, or have like, you know, the connection, to establish this connection. So yes, I think like this is interesting because I completely like, I just got so used to it, having it everywhere. So I just completely forgot about it. So that's the answer.
Yeah, it caught my attention. The other question I was sort of thinking about, and I think I got on Suzanne Anchor when she gave a talk here too. So this is towards Jude. Where you were talking about how like cooking in the kitchen is different than when you're in a science lab, and that science production is different from cooking. And you know what, I actually, when I work with my students, I'm always like, science is just a protocol, it's just like cooking. So I sort of want you to expand upon like why is like that heritage of cooking and moving forward there, like what's the similarities and differences from working in a lab and building in that sort of environment?
I don't actually think that they're that different. I think it's more so the institution that makes it different. Like being in your home space or in like a domestic kitchen space, it's the differences between like the domestic versus public or private versus public. And even like lab spaces are inherently very private. They're not, and I'm generalizing at this moment, but lab spaces or scientific lab spaces tend to be much more private than, you know, just coming over, like me coming over to your place and just like making a meal together. There's not that many bureaucratic and political implications and layers to me being able to access a lab space versus a kitchen space. And though the methodology might be very similar, and there's often some like crossover between some of the technologies or like the things that you can do at home that mimic a lab space or vice versa. I think it's just like the politics of the space that makes it different. But I think if you strip down what it is that you're doing, they are inherently similar. And that's why I find comfort in being in a lab space, even though I say this all the time, I'm not a scientist. I'm an artist first and foremost, but I'm someone that's very driven by their curiosity around the sciences. And that's why I like working in a lab space, even though half the time, like, I don't know what I'm doing. but I like that. And that's also how I like navigate my way in the kitchen. Like I make really good food, but I don't know what I don't know what my end goal is half the time. I'm just kind of like, you know, throwing in a little bit of this, or like I cook by feeling, if that makes sense. It's like a very intuitive approach. And I find myself doing a lot of that when I'm in the lab space, which I don't think a lot of people tend to do that.
No.
So, like, you know, I don't know how to, like, there's obviously some similarities there, but there are differences too. But I think it comes down to just like the politics and the institutional space of like a science lab.
Yeah, I think it's really interesting to think about it like that. I think like scientists themselves, like we don't always know what we're doing either, right? Like we're troubleshooting just like you do in a kitchen, like, oh, this tastes like crap. Or like this doesn't work this way or that way. And sometimes it's a feeling and you go that route, but you take notes on it and you work around it. So I always think they're super, super similar, but I think you're right that it is institutionalized in science where it's not so much in your home kitchen.
Yeah, I wanted to mention also, you're talking about like the kind of like the politics and the accessibility of the lab, thinking also about field work. And I think that maybe Laura can speak to this a little bit too with her field work. And I mean, Daria going out and looking at the mushrooms and collecting the mushrooms, about doing science work in some of these public spaces where it's not within the confines of a lab, and also where there is some of the blurring of the edges a little bit, where you're out living potentially with your collaborators, spending time out in the field, but still having that lab component also as an important part of doing the research.
So one question that I had too of the 18, I think it was 18, but it was a lot, was for Laura, where I was thinking about with the water testing, and I wonder if you were thinking about this with the microplastics, whether population and more important, like social economic sort of conditions around that water, does that correlate with a higher incidence of microplastics? And is that sort of a rationale why you chose those two different lakes? Because I always think of like Skinny Atlas Lake as like the cleanest lake in the US. I don't even know if it's cleanest, but it's very clean. So is that something that you keep in mind with your research?
Yeah, definitely. So we chose a lot of the sites based on how like rural versus, you know, highly developed they were, to kind of have that comparison, because more than likely a lot of the waste is from like incorrectly managed waste, street litter, stuff like that, that can end up washing in, and like impervious pavement and stuff like that will definitely impact it. I think it's interesting that you brought up the socioeconomics too, because for another study I'm doing, I'm just looking at how much waste people produce in their home, like individually. And that is also very different based on economic status. So high economic status countries tend to produce more waste than low economic status countries. And those other countries they tend to produce more compostable waste, which I think is interesting, more like food waste, versus, you know, countries like the US, we produce a lot of like paper waste, plastic waste, things like that, that tend to be mismanaged. I just think it's really interesting to think about like what goes into that, what kind of structures are at play, and just the access people have to different packaging. Like maybe we have more packaging here and that's why, but it is just interesting to see that and see how people, where their waste comes from and what they think about it, I guess. But yeah, that's definitely something I've thought of. Flow conditions are also a really big indicator with plastics, I think. High flow events tend to mobilize a lot of plastic waste into the environment. So that's another factor. But yeah, for sure, those lakes were chosen because Onondaga Lake is so surrounded by developed areas, and then Skinny Atlas is, you know, much more rural with agriculture, potentially like entirely different sources of waste. But I think it also goes to the fact that a lot of developed countries ship their waste out. So even though in some countries, like the people there might not be producing most of the waste that they end up finding in the local environment. So I think it's so different depending on where you are and what kind of sources you're looking at. But yeah, focusing on here for sure, that's definitely probably an impact, but we'll see.
Yeah, thank you.
Anyone else have questions?
I think there is a question from the audience. Let me just open here. I think, and then I have questions, but it is from Pavel Malchevsky. He raised his hand, and I have a lot of questions, but I'll just ask afterwards.
Yes, hi, thank you. So my question is for Daria, and the question is, if I understand correctly, the technology that she is developing and also the theory behind it will help humanity to get rid of or like reduce drastically waste, like plastics and other harmful substances in the world. So my question is there a risk that mycelium or mushrooms will start eating what they're not supposed to eat and like will start destroying the world in the places where we were not thinking? It's like the artificial intelligence, and they have, you know, futuristic movies where artificial intelligence go out of control and like destroys humanity.
Right. Okay. Well, that's a great question. I mean, the thing is, I just want to do like some correction. First of all, I mean, the thing is, the technology I'm using, like, I didn't really develop it. You know, I'm just like using bits and pieces from other open sources, and just kind of, you know, combining this to create to build something that I've been looking for a while. But on the other hand, it's kind of like mycelium and mushrooms, they have been around for billions of years, you know. So, and the thing about, there is actually indication, there's two species, I think, so far, maybe even more now, that have been found, one in Pakistan and one in Amazon, that could decompose potentially plastic, right? So there has been studies, they've been discovered, and they're thinking about implementing the species under the waste, under landfills, so they can potentially help with, you know, like with the degradation of the waste, which is very interesting. But in terms of decomposition, the mushrooms will eat something they're not supposed to eat. I think it's like, I'm not a mushroom specialist, but so far are they decomposing what they're supposed to decompose in? And I think it's like a very balanced ecological system, right? So we wouldn't be worried about this so much. Like the species that I'm collecting, they are all over, and the materials that I'm using are very simple materials. And this is the point of the whole project, to have the most simple, accessible materials to be used. And because I was offered to use something more like chitosan, right, from the lab, in order to understand, like, for the building properties or anything like that. But I specifically stay away from anything that I don't know about, and I stay within the realm of something that it's accessible to everybody, right? Or something that we use every day, things from the kitchen, things from the trash, things from all over the place. So it's highly unlikely, unless, I don't know, we get invaded by some giant mushrooms from space, which is also possible, you know, because in the view of the recent events, right? So it's kind of like a possibility now. Very small one, but it's possible, I think. So yeah, I think it's, and there is companies actually like working, the beauty of the mycelium, the fact that you don't have to invent anything. This organism has been, again, here forever, and it decomposes. There's different species that decompose different things, like there's decomposing cellulose, there's other decomposing other stuff, and we actually know like tiny fraction about all, like some of them right now. So I hope this answers the question, right? All right, thank you.
I'm raising my hand. But if you have a comment, Tom, you go first, and then I'll ask.
No, I'm asking if you're ready to have your questions.
Yeah, I just it's a short comment to the things that are that the mushroom can eat and are in a, not appropriate to be eaten. There was an image of Alice in Wonderland that is a lot of mushrooms on top of it, growing on it. So I don't know. I was just thinking about the visual representation of what is appropriate to eat and what is not appropriate. I don't know about Alice in Wonderland. Maybe this is a question. Shall the mushroom eat Alice in Wonderland or not? But so all my questions. This is actually amazing photo. It is a book that is very old, and these mushrooms, they look like very stereotypical mushrooms. They have a little leg and a hat on top of it, and they grow out of the book. Anyway, so all of my questions, all of my other questions are related to visual representation of all of the participants, because I found all of the images that they have been sharing really compelling. I think the image of the last presentation, the images are also really interesting, but they kind of move towards sound. But anyway, I have a lot of questions, and I'm not sure that I should be taking too much time of asking them. I didn't write them down because I'm actually recording the whole thing, and too much of a chat I think is not very visually compelling. But okay, so my first question is actually to Laura. Where did you take the bug from? This image, when I looked at it, it was so, and maybe it's not the, I mean, where did you get this image from? It is really interesting to look at it and to maybe look for plastic, or maybe think of other things that are on the image. It was really something that I would like to look at for a long time.
Oh, thanks. Yeah, that was actually, I had come across it in an Onondaga Lake net sample. So that's, you know, after the processing and everything to get it onto the filter, and hard bug bodies tend to stick around. So me as a bug phobe, I'm not the happiest when I see that stuff, but I thought that one like really stuck out to me, and it was pretty cool looking. So, but that's under the microscope, so it's enhanced and everything.
How did you pick up the image? Why did you choose this one as opposed to something else to show in your presentation or to use as a research image?
I think it's an interesting one for scale, because you have the bug there, so kind of a sense of like how big everything is enhanced. But it also just shows how dirty the samples can be and how challenging it can be to kind of separate out like what's synthetic and what's plastic versus what's natural, because surprisingly stuff looks a lot alike. I just started the process of counting plastics, and I'll be going through a sample and see something that's a yellow film, and I'll think, oh, for sure, that's a piece of yellow plastic. And then I'll keep going through the sample and see it everywhere and notice that there's like little cell structures within that, and that's probably a bug carapace and not a plastic. So I have to kind of, you know, check things as I go, but even, you know, natural fibers look very similar to microfibers. They just have very slight differences. So I just thought it was an interesting kind of comparison between like what's natural and what's synthetic, and how it can be difficult to separate the two.
Yeah, so there was another question about, or rather comment. I have friends who are chemists, and they do plastic. This is what they do. They just make plastic. And so they're very almost offended by the whole thing about not using plastic. And Tom raised this question previously about collaboration with your subject, or thinking about your subject as something that you can live with. So did you have any encounters with people who would say, why are you doing this? We are making all these plastics to save the world of hunger or whatever. There are different reasons of why plastics should be used, or why plastic should be used in space, in outer space, or other places. Did you have any encounters with chemists who would...
Yeah, so I purposefully chose to include a polymer chemist on my committee for my dissertation, and he kind of presents that balance, but he's from ESF, and he is awesome, and he told me during my candidacy exam, he was like, you're gonna be loved and hated by polymer chemists and, you know, environmentalists. So I think it's really good to have like that dual perspective of what do, you know, other people think of the work. And plastic, I don't view it as a bad thing. I think it's just used poorly. And so I think there's this balance of not going too far in the direction of seeing it as, you know, it's evil and people demonize it a lot. But I think there's that aspect of, you know, it's efficient to make and it reduces food waste. And there's all these other intricate pieces to it that I think if we chose to just replace plastic immediately and not think about those talking points, we would probably make a regrettable substitution, and we would pick something that's probably going to have another bad impact. So I think he keeps me balanced in thinking about that, and people that advocate for plastics do too. And I get where a lot of people are coming from, so I try to take it in stride for sure.
Yeah, my last question was about your interest to graphic design. It seems to me that you're maybe collaborating with graphic designers, or you yourself, you're producing a lot of the graphics that you're using in your presentations, but also some of the graphics which are on your website. I don't know if I can formulate the question, but it was very curious to me. Do you think that a good graphic design or the graphic representation can communicate more of what you would like to say, and how? I don't know. Just share something from your experience.
Yeah, for sure. I actually originally wanted to go to college for graphic design, but I was worried about sitting for too long. So I went into science because you go outside more and do that stuff. But yeah, I'm really interested in graphic design and how scientists can use it to kind of enhance their communication. And sometimes I just feel like there's something you want to say, but it's really hard to say it shortly. I mean, that's a terrible word, shortly, but succinctly. And I think just a picture can say 1,000 words. So I like to play around with, you know, different graphic design elements and design different things. I'm even working on like a low waste cookbook that I'm graphic designing for. So I just think it's a really fun way to bring like art, and it's very soothing to work on your science, but also work on different designs to present that science in a new way. And yeah, I think more collaborations with like art and science would really help people, I think, make connections that they didn't think about before and make it more interesting for them at times, because even though we'd love to say every science talk is amazing, some can be a bit dry and word heavy. So I think adding in the visual aspect definitely helps.
Okay, as I said, I have many questions. And I don't want to, if there's somebody who wants to ask questions, I still have them and I still want to ask them. You know, if someone wants to interrupt, but my next question, I will combine it for Jude and for Daria. And it is about the visual representation of ideas. Of course, this is a question that artists face all the time. And I'm thinking that for both of them, it comes from, both of them are not born in United States, so it comes from a different cultural background, and it is mixed with their presence in the United States, and transnationalism in a way, transnationalism in understanding of images. So for Jude, it is a question about the if actually, have you thought about combining your work with architecture? Because my feeling is that a lot of this, it's not only kaleidoscope, it's a lot of the ornamental or ornament.
I'll tell you a few things that I have on the go right now. So in terms of, like, I'm very interested in intervening with existing architecture and spaces. So like one of the examples that I showed was what I did at the Ontario Science Centre, where I was playing with textiles and softening the place and sort of intervening that way. But I've also been printing some of these patterns in addition to the wallpaper. I'm also testing out printing on tile and just like literally like tiling spaces with some of these patterns that are that originate from everything that you saw earlier in my process. So I am curious and interested in that type of materiality and embedding my work more architecturally into spaces, and thinking also about, again, like conceptually, when I think about home, I also think about like mobility and like a nomadic existence versus something that's like more permanent or like grounded. And so that becomes a really interesting thing for me to explore through materials. So I'm also recycling a lot of the sheer textile that was used in the installation at the Ontario Science Centre to make like this really big Bedouin tent installation. So again, thinking about this like home structure that is, you know, neither here nor there, but then also echoing it with maybe making a structure out of tile, or, you know. So yeah, I've definitely been thinking through a lot of these things, and I'm working through that.
Mm-hmm. Yeah, because it seems to me that Islam culture is actually it's actually very iconoclastic. It is not representational in terms of the subject. It is a lot of ornament that carries all the symbolic that can be carried by images that are representational. And with this, I want to transfer to Daria, because I was thinking this cup. I understood the connection with the China factory and the vessels and stuff, and that probably this is a relatively easy sculptural form to print. So maybe this was one of the reasons to be chosen. But then you were fascinated by the glitches, and these cups, they looked like Medusa, just like some kind of creatures that would represent, maybe if we think about Greek mythology, Medusa is not controlled, and then it is controlled when it is decapitated. So I was wondering if you're thinking of other types of prints that are not cups.
Oh yeah, this cup is just because, honestly, I usually work with more kind of anamorphic type of, like my previous work, I did a lot of sculpture. It's like all these organic forms. So for me, this was sort of like a step completely in a different direction. But I thought it was important for this project because it's about order and control versus chaos, and how even the machine can take over and just start making this like intricate forms on its own that will never be replicated. I am like super interested in creating all kinds of forms. And to be honest, it took me a long time to persuade myself that I needed to print this cup because I needed to address this site. So, because there was so many times I was just like, no, I want to print something else, something more like, you know, because on the other hand, it's like you think about, this is just a cup. It's kind of boring, right? But then when I started looking closer to it, it has all these layers. And then what is the notion of why would it be boring, you know, like, and it's like, and then there is a glitch in the system, and then it immediately becomes this like incredible transformation, right? It's almost like this kind of like a mutation happened, like an aberration in the code, and then it just created this beautiful piece because I saved all the mistakes, and I'm planning on actually firing them into the kiln to create this set of distorted cups, you know, like they're completely dysfunctional, but also very beautiful. And this printer, I mean, it just took me such a long time to like figure it out. It was just a journey, and I just now finally kind of relaxed a little bit. But the applications of this work is just, there's going to be so many, a printer is like a part of this series, and it's just a vehicle, a vessel to deliver whatever the shape and form I can possibly think of that could address specific sites, right? And it's very important, the form is very important for me since I'm a visual artist. And I'm wondering, like, in a way, mushrooms are like a really big thing in my culture, like a huge, every wife knows how to pickle mushrooms and collect mushrooms, like in Russia, it's like crazy. And so I sort of started researching a little bit into like folklore and like old Russian folklore and like shamanism and like Siberian shamanism to figure out like was there specific patterns or something, because it's also about like the order is also about the patterns, right? There's a connection, there's an actual network, mycelium network, it's also a specific pattern. It's like, it's not a fractal, but it's like an approximate fractal, right? So whatever that is. So yes, there will be more crazy. I just feel like if I let myself loose, it's going to be like complete insanity in form and shape. And I wanted to have a solid ground before I start going into complete, you know, wild spree of the imagination. But yeah, there will be way more crazy forms coming.
So my my final question is to Julia, and I was really curious to see where you're going to with your question of what a song, what is the definition of song, and where exactly you're getting it from. Because I think there are a lot of definitions. There's some definitions from people who produce songs, and they may not be rigid enough to be applied to something that is a constant definition. I don't know. It's just interesting to see where you're looking at. That's one thing. The other thing is related to indigenous cultures, and I was wondering if you have looked at some myths about what the whales are doing. Maybe it's just like some crazy ideas of why they are singing, or what is the song about, or I don't know, something. And then suddenly it may become a scientific discovery after being somehow proved. I was just I'm asking this because recently I read, it was not very recent, but I read an article which is about the Stanford research, which was proving that women after giving birth, they need at least 1 year for recovery from giving birth. And I was thinking, why do you need a scientific research to prove this if you just feel it? And this is what I'm saying this not to question the importance of science. I think it is extremely important, and we need the authority of science. But I'm also thinking about the fact that cultures and not culture, like arts or even myths or folklore can sometimes help trigger some ideas of why things are happening, and discoveries, of course. So I was wondering if you have looked at some indigenous ideas of why whales are singing.
Yeah, so, well, I guess I'll start with the question of song. So the reason that I am asking this question of what is song is because another part of my thesis is looking at which mammals sing and trying to trace that evolutionarily to see when did song evolve, or did song evolve multiple different times among mammals? And so is that something that, you know, for humans, is that something that we share with other primate ancestors, or is it unique to humans? And so for this research, I really need to come up with some kind of working definition of what song is in order to study it that way. And I think it's really interesting because we've described bird song as song and whale song as song, and there's a lot of similarities people draw between those animal sounds and human music. But the question of what those similarities are, and also what fundamentally it means for something to be song, is something really complicated, but something that I'm going to be struggling with a lot throughout my thesis. So that's why I started to bring that up there. And yeah, it's something I'm going to be working on a lot more. So if anyone has additional input on what makes a song, I'd love to hear it. But then moving into your question about indigenous cultures, actually something really interesting is that I read that for Hawaii specifically, there isn't a lot of, and this is just based on what I've read so far, it's something I definitely need to do more research on, but there isn't a lot of record in terms of folklore or history of the humpback whales or whales really at all, which is really interesting. And so I think some people have wondered whether Hawaii as a breeding ground is relatively recent, in the past century or so, and that potentially the whales were somewhere else before, and that's why there isn't really a record of it. So that's definitely, I mean, on its own, really interesting if that is an indication of the history of where the whales have been. I know that there is definitely, in terms of particularly like in the Arctic, indigenous cultures where a lot of what we know, especially as it relates to conservation in terms of how big populations were and what their behaviors were like, that a lot of these indigenous cultures that have been hunting whales for a long time, that's where we get a lot of our knowledge about historically what these populations looked like. But in terms of the song, it's not something I've done a lot of research into in other populations, but it's definitely something that is really interesting that I think I'd like to look more into.
Well, I think it's really interesting to think about song as a form of display. You know, in the same way we can look at art as a form of display, and then there can be lots of comparisons with how other animal species display or how plants display or whatever. And I think the whole idea of song and the organization of sound in pattern lends itself to all kinds of analogies, including the introduction in music of noise, right? Which I thought was, you know, so interesting with the micro printer that Daria was just bringing in this whole element of noise. You know, these what normally would be a ceramic cup just became this very funny ice creamy kind of weird substance, you know, and it would go haywire, etc. But I think that I think that the song stuff is really interesting. Songs are very important, you know, just the repetition of patterns of things in all of the presentations tonight. They were all songs.
Thank you, Tom.
I think this has been a great session. I think it was really a wonderful event, and just to see, you know, the 4 points of intellect, you know, kind of forming into a whole, and I hope the audience has appreciated it, and I think it was magnificent.
Thank you. It seems that we're kind of finishing our discussion. If there's some more questions, maybe we can have more. Yeah, great talks, work by everybody. You can read your chat if you haven't. All the presenters, there's a lot of compliments here, and I think it was really amazing to listen to your talks and to see you. And I just want to welcome, and Heidi will hopefully say something afterwards, but I just wanted to say that we are really looking forward for the next part of the symposium, which will be on August 20th. And Matei Vakula is here, who is one of the presenters. He's also a PhD candidate at the Rensselaer Polytechnic. He will probably be graduating next year, hopefully, I don't know, or this year, I don't know, but next year. So he's one of our presenters on August 20th.
Yeah, and we'll hopefully get a couple great scientists that will be presenting. So stay tuned. We'll work hard on that. And yeah, I'm really looking forward to August 20th. This was great.
Yeah, thank you, everybody. I want to thank all of the presenters, and thank you, Tom. And I want to thank all of the students who participated in the organization of the event.
Bye, everybody.
See you next time. Bye.
Guys, thank you.